So today, I'm in the algebra and geometry part of the afternoon. So I will focus my talk. I'm very interested in the role of geometry, how geometry of a cell shapes its function, and how the activity of the cell shapes its geometry. So this is the geometry I'm going to talk about today. Okay, so the first thing I wanted to present is a model for short-term synaptic plasticity. So what would be the motivation for this? We have, in this example, what you observe is two different types of cells, of neurons. On the left, you see the climbing fibers here, and on the right, the parallel fibers. Those two cells are projecting on the same cell, the same neuron, which is called the Purkinje neuron. So the climbing fiber and the parallel fiber are projecting to the Purkinje neuron. They are the geometry of the presynaptic terminal in the two is extremely similar. They have the same size, they have the same number of vesicles, the same maybe organization or number of vesicles ready to release neurotransmitters. But they have very different behaviors in terms of short-term plasticity. So what would be short-term plasticity if you excite the neurons, the climbing fiber, twice within a short time scale? Here you have a 20 millisecond pulse. You will observe in the climbing fiber what we see as depression. So you have the first impulse that will lead to some vesicular release and some extra postsynaptic current, and then the second excitation, the second pulse, will lead to a smaller pulse. This is called synaptic depression. And if you look at the parallel fiber, it's showing exactly the opposite. It's showing some facilitation. So after the first pulse, you would have a current that will be generated, a postsynaptic current that will be generated, and 20 milliseconds after, the second pulse would be bigger. So the question here is, how come those two synapses that look super similar in terms of geometry and organization and that do project on the same neuron, how come they express those very different behavior? The one is really expressing facilitation and the other one is expressing depression. So this was the motivation of this first study, and to start the model to understand what could be the reason for such discrepancy, maybe I can start with a small background about the functional organization of chemical synapses. So from those of you who are not super familiar with presynaptic terminal, the way it's supposed to work at, let's say, glutamatergic synapses is the arrival of an action potential at the presynaptic terminal. So this is the end of the neuron, the one that is projecting the information to the next one. When the action potential arrives, you have here down at the active zone. This is a specific zone that, as you see, is located in front of the postsynaptic terminal. You have here voltage-gated calcium channels. So those calcium channels would be closed, except if you have a shift in the voltage. So when the action potential arrives, you see a shift in the voltage in the membrane, and this creates the opening of those voltage-gated calcium channels and the entry of calcium ions. Those ions would enter, they would probably diffuse within this terminal. But if enough of those ions are binding on the vesicles that are already attached to the membrane, let's say 4 to 8, it's not yet extremely clear, then this binding of 4 to 8 calcium ions on the vesicles that are already docked at this particular part of the membrane that is called the active zone that is facing the postsynaptic part, then this is going to trigger vesicular fusion. This is the way, like the classical model for vesicular release at presynaptic terminals. Okay, so we wanted to build a model for this and try to understand what could be the parameters triggering such a difference in response to stimulus in short-term plasticity. So if you zoom a bit onto the docked vesicle here, what you observe is that these synaptic vesicles that are docked, attached to the membrane on this region that we call the active zone, which is active where vesicles have to dock, and you have all the machinery and the proteins necessary for synaptic transmission. When you have those vesicles, they are docked to the membrane of the presynaptic terminal through a complex of proteins that is called the snare complex. And this snare complex is related to another protein that is called the synaptotagmin. And this synaptotagmin is the protein trapping the calcium ions. And if enough synaptotagmin has trapped enough calcium ions, then those snare complex are going to change conformation, and then the membrane are going to be fused, and you will get the release of the neurotransmitters that are inside the vesicle. So if you look at this scheme, what you see is that this snare complex is supposed to be located underneath the vesicle, right? It's really those transmembrane proteins, and when the vesicle arrives close to the membrane, then it binds. And you have those calcium channels that are entering also, these calcium ions that are entering through calcium channels somewhere in the membrane. We know that those calcium channels are in the active zone, but what we don't know is how much do they colocalize with vesicles. And this is something that is still not yet understood well in the community. So the idea we had here was that probably reaching the snare complex for a calcium ion is not as straightforward as if the synaptotagmin was like a buffer that was in the middle of the terminal. So we wanted to see how much the geometry of this docked vesicle would interfere into the dynamic of the binding of ions. Oops. So to formalize this problem, we choose to consider the narrow escape problem in a cusp. So, which means that we're going to describe the Brownian particles are calcium ions, and we're going to take a look at the first time for these calcium ions, this Brownian particle, to escape our domain here through the red boundary. So the blue would represent the presynaptic terminal, the green would represent the vesicle, and the red would represent the snare complex. Let's say if you're close enough to the snare complex, we consider that you're trapped by the complex of vesicles. So you're going to get mean first passage time in the sense that you have a calcium ion starting somewhere inside this domain, the Brownian particle starting somewhere, and you want to know how long it takes for this particle to reach this small domain. And this is the first step of triggering vesicular fusion if you have enough of those calcium ions doing this. So the formalism of the narrow escape problem is very good for this question. And we know using Ito's calculus mainly that the mean first passage time U is the solution of the mixed boundary value problem that is described here. Dilapation U is equal to -1. You have a reflective boundary condition everywhere except at the target site, the red ribbon here, where you have an absorbing condition. So we formalize the problem this way. And then to go quickly on the method, the idea was that if you consider this problem in symmetrical coordinates, you have a symmetry around theta, right? So we can reduce this to a 2D problem. And in this 2D problem, we can now try to think how can we simplify this problem, get the solution? And the idea here was that we were expecting that the time it takes for a Brownian particle to reach, let's say here, to reach the red boundary here would pretty much depend on how far you are on the circles here. That would be all the circles that are tangent in 0. Okay? It's probably not where you are in X or in R or where you are in Z, but probably where you are at least at the first order on the circle. So if you imagine all the circles here, you can probably understand that the mean first passage time to reach the red ribbon would be the same everywhere on those circles, depending on how far you are from the circle. So this idea motivated the use of the conformal mapping method, where we transform all those circles into line. And the idea was that maybe if we get lucky, then at least at the first order, and which is only at the first order, the mean first passage time should not depend on when you are in T, but only on where you are in S. So you transform the problem, at least at the first order, on a 1D problem. So we did the calculation, and using the matching asymptotics, we find that this is true. And then we ended up with a first order ODE to solve, and this is something we're able to do. And we could find some solution for this problem. So here is the solution U of RZ. So you're on symmetrical coordinate, right? And theta it's independent of theta. And this is the kind of solutions you find. So here you find the fact that you are somewhere on the circle, and if you are close enough to the boundary, so when R squared plus Z squared is exactly equal to this boundary, you end up getting 0, which is the time it takes to reach the boundary is going to be 0. And if you fall, you see that this represents where you are on the circle. So we could do this analysis for the 2D domain and the same for the 3D domain. And if you look at this analysis, what you probably recognize is the curve that is here, where you have, when you're far from the boundary layer, you have a constant solution. And when you are close to the boundary layer, then you see a huge drop in the solution that goes from a value that is super high to 0. So this is exactly what we were expecting, because we know that most of the time for the mean first passage time to a small target, the mean time to reach the target, if you're far from a boundary layer, is constant. And then the constant here would be given by in 2D square root of 2r times omega over d square root of epsilon. And in 3D, the omega over 4 pi d square root of epsilon. Okay, so this is the shape of the solution. So what does this result mean? It means that if you're far away from the domain, from the boundary here, this term is going to be extremely small, okay? And this term is going to be the leading order term in square root of epsilon. So the time it takes for a Brownian particle that starts somewhere outside of the boundary layer is constant. This is what you see here, and is given by this formula. Epsilon is the size of the absorbing window. The 3D domain is the same. Sorry, there is no square root here. This is a mistake. This is 4 pi d epsilon. We could check this versus Brownian simulation, and it gives excellent results, as you can see. Okay, so we had this idea. Maybe the first thing is that we could compare these to other mean first passage time in other geometries. So actually, what we understood here is that like the time it takes is in 1 over epsilon, where the surface of the hole is in epsilon at the power 3.3 over 2. So the surface of the hole would be the surface of the small ribbon. You had already known results, like, for example, if you want to try to reach a small hole in a sphere where the sphere is of radius a, then this is the mean first passage time you get, where the surface of the hole would be like pi a squared. This is just a circle. You can do the same, like the results in the funnel shape curve here has been also derived. This is something that you can use probably when you do like meiosis and mitosis for the cell division. This is a kind of geometry you can find. And again, the mean first passage time, it has a different dependence in the size of the hole. So what if we put everything together here, so what we get is that the mean first passage time from a small hole at the first order is constant. Actually, what has been shown is more than that. It's that the law of arrival is exponential at the first order. Okay, so what it means is that this mean first passage time is integrating the effect of the geometry in the rate, but you can then consider using this rate that what you have is the arrival of your particle is a Poisson process. So this is a way to integrate a very complex geometry into a very simple tool, very simple model. Okay, so that you can consider the arrival at the first order, the mean first passage time is independent of the position if you start outside of the boundary layer. You know that the law that is associated to it, this is something that has been shown, is exponential. So what you get in the end is a rate that captures completely the geometry, and that gives you a Poisson process for the arrival time. So this is the most important and most interesting probably results you have with this mean first passage time at small holes when you're trying to do modeling and multiscale modeling. Okay, but if we were interested in the model of the full terminal. So what we know now is how long it takes for the calcium that is far away to come back. Okay, but what we don't know is what's happening at the active zone. So let's say the active zone, usually model it this way, okay, with several vesicles on a square lattice. And the question of where are the active, the channels is a question that is always under debate, and it's probably different depending on what kind of synapses you're looking at. So the thing is, and this drawing is maybe easier to understand. The thing is, our calcium ions is entering inside the boundary layer, actually, or might enter inside the boundary layer. So now that we resolve what's happening outside, we have to also try to resolve what's happening inside the boundary layer. So we have already the shape of the solution inside the boundary layer, at least at the first order. But what we're interested in is now the splitting probability. So it means like when you enter the boundary layer, when you start somewhere inside the boundary layer, what is the probability to reach the target before getting lost inside the terminal? And this is called the splitting probability. And actually, the PDE that is solved by this problem is kind of close to the one we had before. So using the previous analysis, we could find an approximation of this probability. So we found that the probability takes this form, and we could estimate A using Brunel simulation, and we end up with the approximation of the splitting probability being this one. Okay, so this is the probability to enter the boundary layer before getting lost inside the terminal. So now if we want to integrate all of these things in a full model, the main problem we're going to get is that inside the bulk here, what we have is a classical dynamic of calcium binding on buffers and pumps and whatever. And this classical dynamics is going to be represented well by deterministic equations, right? This is let's say the mass action laws to be super simple. Whereas here, the entry of calcium inside the active zone through the voltage-gated calcium channels and the binding or not binding on the vesicles is well represented by a stochastic approach. So we choose to couple the stochastic approach to the continuum through this Markov model coupled to mass action. So the idea was to represent with continuous equation the bulk, and with a Markov chain the target, the binding to the target, the discrete and the stochastic part. So, and we could do this thanks to the mean first passage time theory and thanks to this arrival rate of the Brownian particle to this snare. That's thanks to the fact that this is an exponential load and that we could represent this as a Poisson process. Okay, so then you do the simulations and you can observe the distribution of release time or uniform channel distribution. So what you can see, I did different simulations for different number of buffers, okay, that bind the calcium. So for a different shape of the calcium impulse curve. And what we could see is that this calcium shape is actually triggering the vesicular fusion. So after calcium entry, you would see a huge peak corresponding to vesicular release. And then if the calcium stays high in the cell, what you would see is a lot of vesicular fusion happening after that due to the ions coming from the bulk to the active zone. If you lower down the calcium, you will lower down this second peak. And if you don't have any calcium, because you have a lot of extrusion or buffers, then you would not see this peak. Okay, this vesicular fusion. So this was a first hint into what's happening in the synapse and how controlling the level of calcium inside the cell, which is something that is extremely complex, and that the cell is paying a lot of energy doing it, can control also vesicular release. So a lot of one of the main idea of this is one of the main idea of facilitation is that it's coming from higher calcium inside the terminal. So it's not exactly the way it's been described, but what we see is that this is true, that the higher calcium in the terminal is going to trigger a lot of vesicular release inside the, like, not that directly because the calcium is high, but because you have all this process of coming back and probably like synaptic priming, et cetera, et cetera. So what we wanted to investigate at the beginning, and this is probably, I will not have time to go further than that, is that what we could see by changing some parameters in our full model is also that depending on how you could, on some properties of your SNAP complex, so the complex of protein that is attaching the vesicle to the membrane, depending on the dynamic of this complex that is not yet well known and it's under debate, you could, with the same calcium, calcium dynamic, obtain very different release time. Okay, so this was a way to investigate how fast snare unbinding and slow snare unbinding could affect vesicular release. And what we could see is that if you have a slow snare unbinding, so the calcium can unbind for the snare, but slowly, depending on the number of buffers you have inside the terminal is able to make the synapse shift from a facilitating synapse, which you see here with the purpose ratio that is bigger than 1, to a depressing synapse that you see where the purpose ratio is smaller than 1. So, I had other things, but probably I don't have time for it. I don't know, Daniele.
You do have a few more minutes, given that we also started a bit later, so you can take...
Okay. Maybe let's stop here, and if people have questions, I would be happy to find it to answer.
Thank you very much. Then. For a change, let me start from the questions online. So we have a question from Romain Vels. Romain, just perhaps unmute yourself and ask the question yourself.
Hello, can you hear me?
Yes, we can.
Yeah.
Thank you very much. Thank you very much for the talk, Claire. It was very nice. I have two questions. The first one is, can you discuss the modeling of ion channel dynamic of calcium dynamics by a Brownian motion? Do you think it's plausible? And for example, do you have any interaction between the ions, or if you have too many in a specific, for example, if the density is too high, or if you have actin and preventing the free movement of the ions? Do you have any idea?
So it's a huge assumption. Anyway, the vesicle is never that crowded, so that the motion of ions is perturbed, I think, a lot, so that the Brownian motion is not still a good result. So usually people, they measure the diffusion coefficient, and this measure of the diffusion coefficient is already taking into account a kind of crowding. And we are not in the case where you have so much ions that the crowding prevent them from having this almost Brownian motion. The other thing is that it's more problematic when you reach calcium sensor, for example, when you reach the synaptotagmin, because then you know that you have this complex of protein that is charged and that is kind of attracting the ions. So I feel, for me, the biggest problem would be when you're reaching the snare or whatever buffer or pumps. So the way in modeling we resolve this problem, for example, for the buffers, actually the radius, actual radius of your buffer, and if you think about Smolachowski, is the radius that takes into account the tracking.
Okay.
But you get kind of a bigger radius than the real ones, because when you measure this Poissonian rate, actually the fact that when you're close, you can trapped is already taken into account. That's why we choose to have this ribbon instead of having specific sites underneath, saying like, okay, if you're close enough to this snare complex, what's happening is probably too complex to be described with simple Brownian dynamics, and we don't really want to enter into these details. So we just let go. The other thing is that we know with the Poisson-Nernst-Blanc theory that the charges are pushed on the boundaries, so we probably ignore part of these Brownian ions that will be directly pushed on the boundaries. But the center of the cell is supposed to be electroneutral. So Brownian motion doesn't seem too bad.
Okay.
Yeah.
Thank you.
Bruno, you have a question? Yes. Go ahead.
Can you hear me?
Yes, we can.
Yeah.
Hello, Claire. Yes, my question is about the beginning of your talk. You were assuming that the spike is arriving in order to trigger the whole synaptic plasticity process. But as far as I understand, you don't need a spike. You just need to have a rise in the voltage. I am asking that because not all neurons are spiking. In the retina, most neurons are not spiking, and it is believed that short-term plasticity could arise also in the retina. So would it be possible to adapt your model in the case where you don't have a presynaptic spike, but just a rise in the voltage?
Yeah, sure. So I took the standard description because this is a very general model. Yeah. And the idea here is that the molecular machinery I choose to let the calcium enter is the voltage-gated calcium channels. Yeah. And they have a pretty high voltage shift. But the voltage-gated calcium channels, they don't need an action potential. They just need a shift that is high enough so that they open. If you have different types of channels, low voltage channels, whatever kind of calcium channels, you can get exactly the same kind of results. The idea is just having calcium entering. So yeah, it's true that I simplified a bit by saying like action potential rise and the voltage-gated calcium channels enter, but this model doesn't care that much about the action potential. It's just about representing the flux of ions entering.
Yeah, thank you. Thank you very much.
Thank you. So we have no more questions online, so perhaps we have questions in person from here.
Yeah, thank you for your talk. I'm wondering, the first slide you showed, we had one case with the climbing fiber exhibiting paired pulse depression and the parallel fiber paired pulse facilitation. And at the end, you just explained, so to speak, facilitation. And if I get your model right, facilitation is due to saturation of endogenous buffers, so that when the next calcium entry comes, you have more free calcium available or not?
Yeah, I think this is the classical idea, and I didn't like it. I didn't trust it as a student because it didn't make any sense for me that the fact that you have more calcium inside the terminal would lead to, when the second spike arrives, bigger release. Because I was like, the time scales are not the good ones. Like the time it takes for one ion to find back with this Memphis pressure time, it's giving something like 4 seconds. Is it 4? Is it 3? Is it 2? Who cares? It's orders of the seconds, right? Whereas the voltage, like the fusion of vesicles, is less than milliseconds. It's microseconds actually. So it didn't make sense for me that the fact that you have more calcium inside the terminal would lead to a bigger release. So, but this is what I've been observed experimentally. And with this model, I find an explanation that at least I understand and that makes sense. And that some people call like snare priming. So actually, when the calcium is high, you have some of those calcium. So this is Poisson process. So the more calcium you have, the smaller the time it takes for the first one to arrive, right? You divide by n the number of ions. So actually, if the number of calcium is high, you would get some calcium ions coming to the snare. Because you need 4 to 8, you can imagine that you have a first spike, and so you release the vesicles that are close to the calcium channels, and you release the first vesicle. Some vesicles are not released. Some calcium ions are still coming back because calcium is high, so you still have this calcium coming from the bulk, priming the snare. Let's say you have 3, 4, 5 calcium ions that are left from the first spike and from the fact that you have this arrival because calcium is high. Then when the next second spike arrives, you don't need 8 anymore to activate this snare. You need only 3. And that's probably why the probability to release is getting higher. So this was my understanding of what could cause this, the fact that when you have a higher calcium, you're going to get a higher release. This is something that's been observed. So, but this is, and because the unbinding from the snare is not something that is really well known, if you have a very fast unbinding, this is not going to be true. But if you have a slow unbinding, this is something you're going to observe. This was my point, I think, in this. I would have to test this, but I think people have done some stuff in the literature, but yet, it's something we should see with people doing experiments. But my idea would be this one, that you can shift due to this calcium.
Thank you.
Any more quick questions? We have perhaps time for one. If not, let's thank Claire for a very nice talk.
Thank you.